to join them. So in the interest of time, I will begin by introducing our plenary speaker today, which is Professor Laurence Carvalho. Laurence is currently at the UK Centre for Ecology and Hydrology, where he's a freshwater ecologist and is also a visiting professor at the University of Stirling. Laurence's history kind of began by doing a BSc in biological sciences in Edinburgh before moving on to a PhD with the famous Brian Moss and of course Suzanne McGowan, who's amongst us in the crowd. And that was at the University of Liverpool, and that really kind of began his interest in cyanobacterial blooms and other algal blooms. He was for a period also a lecturer at the ECRC in the Department of Geography at UCL, where a few of us also know him from. In terms of his membership of the BPS, Laurence has been a member for over 30 years, which I can't quite believe myself. So really starting from Brian's interest when he was doing his PhD at the University of Liverpool. His research at the moment is on the sustainable management of freshwater systems. So this kind of stemmed from his PhD on the Cheshire Meres when he was at Liverpool. And he's very interested in understanding controls of cyanobacterial blooms and populations. More recently, his work has been very informative and quite pioneering in terms of developing the UK and European environmental standards for phytoplankton and cyanobacteria in lakes, and in relation to the implementation of European...
Yeah, just time to connect my headphones in.
Which is the, excuse me, just while I mute, which is the Water Framework Directive, essentially. More recently, he's also led the development of the UK Lakes and Portal, which is a source of information on over 40,000 UK lakes, and also been involved in developing the Bloomin Algae citizen science app, which I've also used, which is great fun if anyone hasn't. And this is for the public to report suspected HABs or harmful algal blooms, and he'll be using his talk today to tell us a bit more about it. So without further delay, I will mute myself and ask Laurence to please share his screen and give us his talk today, please.
Okay, thanks, Ginny.
Thank you.
I'll just get set up here. Just get my screen shared. So this. Okay, fantastic. Well, thanks very much, Ginny, for the introduction, and also just to the whole crew at University of Nottingham for inviting me and organising this meeting. I think it's really brilliantly organised online. I've never seen such a good online conference, actually, in terms of the different ways we can all get together and talk about algae, favourite topic. So, as Ginny says, I'm going to talk a little bit more about some of the themes she mentioned about new ways of monitoring, sort of innovation in monitoring.
Sorry, Laurence, you've muted yourself there.
Apologies, that might have been my fault. I'm sorry. Oh, Laurence, sorry.
Do I?
My
Okay.
apologies.
Okay, is that okay now?
Yeah.
Right. So, yeah, I'm going to be talking about three aspects of work. One is about health risk monitoring and cyanobacteria health risks. I'm going to talk particularly about two aspects of innovation in monitoring, using citizen science, the Bloomin Algae app that Ginny mentioned, and also satellites. And then using this sort of the increase in data that we now have through both regulatory monitoring, satellites, and citizen science, using that to understand sort of drivers of cyanobacterial blooms. So, moving on. There we go. Right. So, just a bit of background about cyanobacteria. I know I'm speaking to an algal audience, and so you're all probably very familiar, but just in case there are a few in the audience who aren't so familiar with this group, as many of you probably know, they're a very ancient group, one of the first forms of life on the planet, and really without them we wouldn't have oxygen in our atmosphere. So we have a lot to thank cyanobacteria for, and they've also play a very critical role in the global nitrogen cycle as well, being one of the main groups of bacteria that fix nitrogen. So they are particularly important from a sort of global biogeochemical perspective. They aren't just freshwaters. I will be talking largely about cyanobacteria in freshwaters, but they are also present in marine environments, and you see them in a number of forms, so single cells, long filamentous chains, and large colonies. The bottom picture there is the sort of very common bloom-forming species Microcystis, or genus, and that's one of the ones that we have a lot of problems with in UK lakes. I just wanted also to highlight three sort of particular specialist cells that certain cyanobacteria have, as that they are very important to the sort of ecology of the group. So firstly, the heterocytes, and I can use my laser pointer here that George showed me earlier today. So heterocytes are basically specialized cells which have an anaerobic environment that is needed to fix nitrogen. And actually more research in the marine world has shown that cyanobacteria don't necessarily need to have these specialist cells to fix nitrogen. They can actually change their physiology over a diurnal cycle, so that at night time they create an anaerobic environment in the cell, fix nitrogen, whilst in the daytime they're photosynthesizing, have an oxygenic environment, and switch their physiology. So incredibly impressive sort of ways that they can change their functioning. The second main type of cell that is very important are acinites, these resting stages that some particular filamentous forms have. And these are particularly important for overwintering, or when conditions aren't good, they will sink down to the sediments, and then at the right time, often in spring, with increasing light levels, they will germinate and come out into the plankton. And I think it's important to think about cyanobacteria, even planktonic cyanobacteria. They don't spend all their lives in the plankton. They have many of them have a benthic stage, and to really understand risks, we actually do need to understand benthic stages much more, as well as planktonic stages. And then the final cell feature, the aerotopes. You can see the little white dots in the Microcystis colonies here. These are enable buoyancy regulation, and are the reason why some cyanobacteria that have aerotopes are able to, or you sometimes see them forming these surface blooms where they're trapped on the lake or reservoir surface. And these blooms are often the cause of the sort of highest health risks that we, because of the sort of high hazard that they have. As I just mentioned, the reason there's a lot of interest in cyanobacteria, as you all know, is they produce a number of toxins that are very hazardous. So these can be liver toxins. So the Microcystis and Dolichospermum produce these microcystins that can impact on liver functioning. Neurotoxins, there's a range of different neurotoxins that they produce. Cytotoxins that affect cell functioning, and dermatoxins that give skin rashes, and pretty much all cyanobacteria produce some forms of chemicals that cause skin rashes. But you'll see from this table that actually, many of these species or genera produce several of these toxins. So it's not just Microcystis that produces microcystins, but many of the filamentous forms as well. And the same with the neurotoxins, many of the same genera. So you may have a particular species and it can be producing several toxins at one time, or switching some off. And the function of these toxins isn't completely known, but certain, and certainly the understanding of what causes toxin production on and off is really poorly known. And just to sort of highlight this fantastic book that came out fairly recently, a few years ago now, and there's now another 2 in the series, I think, that have just come out as well. So I think I wanted to sort of just put this picture up, or several pictures, to just remind ourselves that when we're talking about cyanobacteria, we're talking about a very diverse group of species with very different morphological and physiological traits, with very different life strategies, many of them, and different toxin profiles. And a lot of my research is actually about generalizing the responses from this diverse group. But it is always important to bear in mind that different genera and different species can have quite varied responses to environmental change, and also can have very different health impacts depending on their toxin profiles. So, as I said, because of these the toxin production, they are an extremely high hazard to both human health and animal health. Many of you have seen notices probably around lakes and reservoirs around the UK or wherever you are attending this meeting. And those are because there are very clear known impacts that can cause severe illness or death in people or animals. And slightly more, well, not more concerning, but of increasing concern, that these neurotoxins that cyanobacteria produce may be responsible, or maybe one environmental factor causing hotspots in motor neurone disease and other neurological diseases. And these hotspots have been seen around lakes in the US and along rivers in Australia. And so certainly there's quite a bit of active research going on now, trying to understand the scale of that risk and whether cyanobacteria are responsible. So as well as being very hazardous, the other thing that affects risk is exposure. And particularly when these surface blooms form and we get shoreline scums as winds blow those blooms to particular shorelines, then exposure can be really increased. And that can be just with skin contact with water, through ingestion of water when you're swimming or eating fish, for example. And animals, livestock and pets are particularly vulnerable in this respect, because on a hot day, you'll often want to walk your dog by some water, and they will be prone to going in, swimming to cool off, drinking the water, and licking any cyanobacteria scums off their coat. And that's why we do see a number of dog deaths each year in the UK. And I guess the big exposure pathway that is really unknown is inhalation through aerial exposure. And there's certainly one of the reasons why that's becoming of more interest is because of the hotspots in motor neurone disease, to understand whether that pathway could be important for people living in and around impacted lakes. So just to sort of summarize, yeah, the health impacts of the chronic exposure over lifetimes through both food or aerial pathways is currently largely unknown. And when I'm talking about food there, I'm talking about, say, for example, fish eating, but also through things now, food supplements, health supplements that aren't controlled so well. And certainly there is evidence in the, again, I've seen from the US, of food supplements containing spirulina and other, actually having levels of toxins that are of concern. Because of these known health risks of cyanobacteria, the World Health Organization does have very clear guidance for drinking water quality and for also safe recreational use of water environments. And there have been various publications coming out of WHO, and they were due to have a very large update in, I think, 2019. That was delayed, and it seems to have been delayed further. I imagine 2020, they had their minds on other health issues. But hopefully, I believe that revised guidance is due out fairly imminently. And I think that will address some of the issues that have come up in the last 20 years in terms of neurotoxins and other aspects and food ingestion through food. So in the UK, the only sort of really detailed guidance we have is from the Scottish Government. Both the health and environment is a devolved issue, and the Scottish Government have much more sort of, and Health Protection Scotland have been taken this sort of issue, I guess, a bit more, where they put more effort into developing guidance for local authorities and landowners. And there's been a number of iterations of this. We actually had a new version ready for 2019 that is still awaiting publication because we were hoping to update it following the WHO guidance. I'm hoping that'll come out this year, whatever happens with the WHO guidance, because it's quite a lot of improvements in it. So just some of the things that come out of these WHO guidelines. So it does list a number of levels or thresholds of cyanobacteria abundance and cyanotoxin concentrations that relate to different levels of health risk. And the one that's used operationally a bit more is this low health risks. So above that, you can be expected to get impacts on skin rashes, stomach upsets, and things like that. But as you can see, any surface scum immediately puts the water body at a high health risk, where there's risk of severe illness and even death. And so that's why surface scum bloom forming cyanobacteria are particularly important, because of that chance of high health risks. The other thing to point out about this guidance is that it's quite old guidance now, and it only has guidelines for one toxin group, the microcystins, and so consequently, most agencies only monitor when they have a need to monitor cyanotoxins. They generally only monitor that one group, and I'll be talking about the implications of that a bit later. So major health risks because of the widespread hazard and exposure to cyanobacteria, but that does leave us a challenge for monitoring algal blooms from a health perspective. And that's because we have, in the UK, we have over 40,000 lakes and reservoirs that are more than 1 hectare in size. And 99% of those are less than a half kilometre squared, and so aren't covered by general regulatory monitoring. And the bulk of those even are less than 1 hectare in size. And so we really are talking about having to monitor a very large number of water bodies in terms of health risks. The other issue is that, as many of us know, cyanobacterial blooms are very unpredictable, as is toxin production, both across the country and also within a lake. So some of these large lakes, like you can just about see Loch Lomond here, you may actually have scums, so high risk events at some points in the loch, but up in the north it may be completely fine and very, very low levels of cyanobacteria. So there can be very large spatial differences within a water body. I think the important thing is to say that, basically cyanobacteria can occur in a wide range of fresh waters of any size, and so really it does require some sort of fairly comprehensive monitoring. And as I said, you can have problems with benthic mats as well as planktonic cyanobacteria. So just monitoring plankton doesn't necessarily always highlight risks. So just an example of what regulatory monitoring goes on. As I said, only or largely lakes above 0.5 kilometres squared or 50 hectares are required for monitoring under the Water Framework Directive. That's about 1% of UK lakes. A few smaller water bodies are monitored if they are high conservation interest. And that's because this is really monitoring for ecological health, environmental quality, not for human health or animal health risks. And at these sites where WFD monitoring goes on, the phytoplankton composition is monitored usually at one edge location, taken 3 monthly samples during the summer, and that can be just for 1 in 3 years or so. So as you can see, it's enough to give you a picture of the ecological status of the site, but it's certainly not necessarily sufficient for health risk monitoring. And in 2020, I don't think any agencies in the UK were able to keep their monitoring programme going due to the COVID risks affecting logistics of sampling. So in terms of monitoring the risks to human health, there's actually no mandatory reporting of harmful algal blooms or of bloom-related health incidents in the UK, and that's apart from designated bathing waters. So if a water body is a designated bathing water under the European Bathing Waters Directive, then it should be sampled monthly during the sort of summer season from May to September. There's only 10 inland lakes that qualify as designated bathing waters, and that's a picture of one of them, the Serpentine in London, which is hugely popular. I think most days there are people swimming throughout the year, and I've sampled that for a few summers when I was at UCL, and I don't think I ever had a sample that had low levels of cyanobacteria at that site. In fact, very high levels of phanosomenon at times. But it didn't seem to close the water body for swimming. Because I don't think cyanobacteria are routinely monitored at these bathing waters. It's E. coli and other pathogenic bacteria. But some of them they are. And I think Serpentine they are, but maybe not frequently. In Scotland, we have a little bit more than that. So we, under the guidance, the local authorities are supposed to run visual surveillance programs of their lochs at a sort of weekly to fortnightly basis during the summer. But then there is also across the UK ad hoc reporting to agencies, usually to environment agencies, by the general public. And then these environmental agencies, SEPA, EA Northern Ireland, Natural Resources Wales, will often do some reactive monitoring, in particular if there have been some animal or human health incident. In Scotland, we do have a sort of an environmental incident surveillance system that reports also any issues to local authorities and maintains a sort of national database of incidents. I think England or Public Health England are in the process of setting one up. I'm not sure if it's set up yet. But if you look at the Health Protection Scotland one, they do produce an annual summary, and cyanobacteria are usually the most commonly reported microbiological incident. So it does highlight their sort of widespread, it's a widespread issue across the country. But it has to be said that the reporting to this system is on a voluntary basis, and therefore it's not systematic. And so even though it is encouraged, bloom incidents are almost certainly under-recorded. Well, we know they are because we have other data on blooms. So just to sort of talk about human health risks, I want to sort of tell you this little story, which came to us in February last year, 2020, just before lockdown. And it was from the World Championships for winter swimming was held in Slovenia at Lake Bled. And it is a lovely site. If you go to Lake Bled, it's a beautiful lake. But the time of the Winter Swimming Championships, it didn't look quite so nice. And there was this very extensive bloom of cyanobacteria called Planktothrix rubescens, which has a red pigment in it as well. Normally found in deeper the sort of metalimnion layers, so deeper layer in alpine lakes, but certain times of the year, certain conditions, it will come to the surface. The swimmers were told that this wasn't the risk and that they could just wash themselves after swimming. But what we got reports back were from several UK swimmers who were there, became ill with flu-like symptoms and gastrointestinal illness. And they contacted us to say, well, is this a problem, cyanobacterial bloom? We followed that up with agencies in Slovenia, and they did say they tested the water a few weeks before the championships, I think 2 weeks before, and cell densities were above the WHO thresholds, 30,000 cells per mil, I think. But they tested for microcystins and they were below, they were undetectable. And so they assumed that there were no health risks. However, that ignores the fact that this species produces a number of neurotoxins as well, so anatoxins and saxitoxins, and they didn't test for those. And so what the symptoms people may have been seeing were associated with the neurotoxin exposure. So that's pretty sort of worrying in itself. But there were over 1,000 participants at this event as well. It's a big event, and there's no official reporting of any health incident either in the UK or in Slovenia associated with this this incident. So it just highlights how we don't actually really know the levels of health impacts that we get in terms of human health. Then if we move to animal health, well, we do see quite often reports, and these are just 3 examples from 2020 of reports through in the press of pretty sad stories of either severe illness or dog deaths in the UK. There's one from Wales, one from Scotland, one from England. The latter one was Windermere. However, that's basically the only monitoring that we see that is through the media reports of and we see deaths of dogs, cattle, fish, birds, and there was a big elephant poisoning this year as well. Sorry, last year. Many elephants killed in Botswana. The thing about this, there's no centralized reporting of bloom-related poisonings. Generally, there's no follow-up cyanotoxin analysis to these deaths or severe illnesses, and so the incidents are always labelled as suspected poisonings due to blue-green algae. And so, like human health, the scale of the impacts on animal health is unknown. It's actually only I have a Google alert service, news alert service set up. So now we are beginning to monitor and get a picture of the sort of number of severe, usually dog deaths that we have in the UK every year, just by using a Google News alert service. But that's obviously likely to be underreported as well. So I think the sort of main sort of point I want to stress is that there is a need to take a more sort of One Health approach to monitoring the algal blooms and their impacts on health. So there is this regulatory monitoring looking at the health of the environment, and there is a number of institutes that also monitor toxin levels. But what we really need to do, and we're beginning to do that in Scotland, much more, Health Protection Scotland are trying to link that up with health incidents on animal health and reports of animal health and reports of health incidents with people. But the I think where I've seen this really now properly established is in America with the Centers for Disease Control and Prevention, and they do now bring together agencies of both human and animal health and environmental monitoring, and created this sort of database surveillance system to look at the whole impact of harmful algal blooms. And some of the early data from these are showing that actually you do often, the sentinels of human health problems are animal health. And so you often see livestock illnesses or pet illnesses and deaths before you then, and then you do see follow-up health issues with the public, usually following those. So it's beginning to show a picture of more health impacts than we originally thought. So I wanted to talk about two sort of new innovations in monitoring that can sort of potentially help with this risk monitoring and risk management, and that's citizen science and satellites. Here at the UK Centre for Ecology and Hydrology, we developed this app called Bloomin Algae. We launched it in 2017, so 4 years ago now, and we sort of try and publicise it every summer for people to use to report blooms. And it's where public can just send in a photo from any suspected bloom. They use the phone to give their GPS location of the bloom and the date, and they can tell us about any activities that are going on at the site. It can sort of help us judge both the use of the water body and the risk to that use. And also with this app, two other things that I think worth pointing out. There is an ID guide, so the users, if they become more regular users, they can start to learn what is a bloom and isn't. But there is some advice on there that you heard in Hannah Kemp's talk earlier yesterday, which tells them, gives them some pictures of what is a harmful algal bloom and what are other things that can be mistaken, like filamentous algae or duckweed. There is also a notification service, so anyone can set up a notification for a particular region, and I'll talk a little bit about the people who have done that so far, but that is a very useful, can be a useful service. So just to highlight a bit of the data from this. These are data from 2020, and you can see a sort of a big close-up of central Scotland there, where we do get a lot of records. And again, I'll say a bit why that is. And what you see, we're getting monitoring data in 2020 from about 151 sites, and of those, over 50% we accepted, and about 11% rejected. So in total, that means that 69% of cases, these sites don't actually need further checking. They've been sort of identified as high-risk sites with the 58%. And so there is actually no need for a sample analysis. The warning signs can be put up immediately just from the visual image. And there are still about a third of cases where plausibles, where either the picture isn't good enough, and here you can see a picture of that we couldn't make a decision on. I'm pretty sure it's probably duckweed, but without a closer photo we couldn't be certain, and so we often do get back to recorders and say, can you take a close-up photo? And many of the plausibles, or some of the unknowns, are also if they don't submit a photo. But anyway, it provides this, as I said, there's an early warning notification. SEPA, Jan Krakowski, who's here, gets a notification in any record in Scotland, and many of the Scottish local authorities have set that up. We also try and get feedback to the recorder within 24 hours, or certainly 1 or 2 days in the summer period. So these are the sort of agencies who have set up a current notification service. So as I said, SEPA, Scottish Water, and then a number of councils across Scotland and the Lake District National Park Authority. And that's largely because of pushing from Health Protection Scotland for councils to use the app to support their monitoring. And that's why you see this really cluster of records from across central Scotland, because not only do the local authority environmental health officers have a notification service, we find they use the app a lot themselves to build up their sort of knowledge of what is and isn't a bloom. And so many of those records across central Scotland are from local authority environmental health officers. And the Lake District National Park Authority as well, we get a lot of records around Windermere from their rangers. You'll hear more about this. I just want to advertise Anne Dobell's talk, who'll be talking all about this on Thursday at 10:45. So do please go and listen to her talk. And just a few stories from the app. So just from last year, this is a nice photo sent in of a bloom in Windermere, but in January 2020. And this is from a dog walking business. And the person sending these photos in now has become a pretty regular user, because it's important for his business. It has a major impact, and he doesn't want to be responsible for dog deaths when he's walking dogs. So what we have found, though, through the app is that we get many more records than we ever imagined in winter periods, particularly for some of these large, fairly oligotrophic lakes around the UK. We've had them from Loch Lomond, Loch Tay, as I said, Windermere, when we really wouldn't have thought you'd have algal blooms, and certainly the population densities in the water column aren't high, but you just need the right conditions for the whole of the population to come to the surface and then blown to one shore. And so in these very large lakes, there are these risks of very localized shoreline accumulations. Another example, more typical, is in summer. Obviously, we get a lot more records in summer, and this was a sailing club who wanted information on whether they should be allowing sailing to go on, and whether they should be putting up warning signs to their club members and to the public. And we do get a lot of really positive feedback from a number of users, just general members of the public, particularly from a lot of wild swimmers as well. We get good feedback, and they are beginning to use it as a service. On request from local authorities, actually, we've now produced this information poster. So rather than, this was actually first Lochleven, they muted it, and Perth and Kinross Council, they wanted to not just have warning signs up, but they wanted to have something that could sort of inform the public a bit more, and also give them a bit of advice on what they should do. And so we have developed this poster that you can download from the Bloomin Algae website, and you can actually download an editable version, so you can put your own contact details on it. Just a few updates on what's coming soon. The data is available from the iRecord website, so it's freely available, but it will be made available certainly for, I think, the English data. I don't know whether it's going to cover the whole of the UK, through a new website that the Environment Agency are funding, along to put a load of their data online, along with a load of citizen science data. And so that's with Freshwater Watch, Riverfly Partnership, and Bloomin Algae will be in there as well. And you can just see a little snapshot. It's I think coming online very soon. I think the most exciting news for us is that there will be a new we're currently developing a new version of the app that's going to sort out a few issues. 4 years is a very old software for an app, I think, now, and so we need to do an upgrade. We'll hopefully have a multilingual version, so you can select the language that you want to use it in, and that's because of a lot of requests we've had particularly from Belgium to develop an app for Belgium. We'd be open to anyone who wants to translate the text into Welsh or Gaelic or whatever, that we could have multilingual versions across the UK as well. And we'll be running a load of app training for local authorities, Scottish Water and SEPA, in the spring as well, using the new app. I guess so. One thing on that, so the big improvement is that they will be able to see a map of records around your location, and you'll be able to see your records you submit. So currently you have to go to the website for that, but now it'll be in the app. And also it makes a photo compulsory, and that's going to sort out a load of issues that we have with some recorders forgetting to put a photo in. And as I said, we're hoping the new guidance will come out of Scottish Government this year, where actually local authorities are really encouraged to promote use of the app. And so we will hopefully see a sort of greater picture across Scotland. In terms of pros, I think citizen science is fantastic for giving us pretty good spatial coverage and high frequency, particularly at these smaller sites as well across the UK. It provides we provide a rapid response to what are high risk events, and we provide this notification service to local authorities and agencies, as well as businesses. I think more important, well, not necessarily more importantly, but I think other value of it is that it teaches teaches the public to recognise harmful algae, and it can empower communities to push for restoration measures at their sites. Obviously, there are some issues with measurement quality. You can only say a certain amount of confidence when you get a photo sent to you. And so this picture here is what I thought was a bloom. It was followed up by the Environment Agency, and actually it was a euglena, a bloom of euglena, and so not cyanobacteria. So I've been learning myself through the app of what is and isn't a bloom, and now I sort of get the feeling I can tell a euglena bloom from a cyanobacteria bloom from the look of it, because we've had a couple of those. Anyway, so there are issues, and there's obviously issues with uncertainty that we can hopefully. We have a number of things that's simple stick tests that the public can do that can help with that as well, so distinguishing duckweed from cyanobacteria. There are clearly issues of sampling bias, and here I'd really like to sort of reach out to our Irish recorders, Northern Ireland and Wales, to please download the app and start sending in records. We have very few records from Wales and 1 or 2 from Northern Ireland, but none yet from Ireland. And I should also say that one sort of con is that it is reliant on volunteer algal experts to verify records, and that's myself, Anne and Jan largely do it for the UK. So then on to satellite remote sensing. I realise I've got to really push on a bit with my time. So this is really work by Stirling University, but they're involved, and I'm involved with this global initiative called AquaWatch to develop monitoring service for chlorophyll in particular, but potentially also for phycocyanin and cyanobacterial blooms, using the new suite of satellites that the European Space Agency launched over the last 5 years. And in particular, there are satellites that, as I said, are particularly focused on monitoring chlorophyll, but also some that are suitable for monitoring phycocyanin, and others that can just distinguish whether there are floating scums or not. And so now we are able to sort of, using this service, record at quite low spatial resolutions, 10 to 60 meters, and fairly frequent, every 5 days or so. At larger lakes, we can actually measure phycocyanin, so with a 300-meter resolution every day, potentially, in certain parts of the world. And there is now a UK Lakes Observatory of satellite images of chlorophyll that Stirling have set up. It's not quite live. I think it's being tested by the environment agencies in the UK, and this has monitoring data, real-time monitoring data or near real-time monitoring data from 933 lakes, and you can see their sort of distribution across across Great Britain there. And the advantage of the Stirling method, it actually looks at the water color and can dynamically select the right algorithm for each pixel, so you get much better algorithms used to measure chlorophyll. And just some of the data from that, you can see this is a time series of chlorophyll A in Esthwaite water in the Lake District for 3 years, and you can see just the huge amount of spatial coverage you get, with thousands of pixels here for one water body, thousands of sampling sites, you could say, and you can really see the development each month and where there are any spatial patterns within a lake. We have done other work at Loch Leven looking at phycocyanin with Stirling, and this is some images that Peter has analysed based on the NERC aircraft, actually not a satellite. But you can really see clearly cyanobacterial blooms along shorelines using some of these aerial images. And some of the new commercial satellites coming out have this sort of 1 meter resolution as well, so or maybe 3 meter resolution. So there are really possibilities are increasing every year in terms of what we can do with satellite remote sensing. So satellite remote sensing gives us this whole lake coverage and also global coverage, so it's really impressive. And the frequency is now daily to weekly. So we're talking about really massive expansion in monitoring capabilities. And it certainly also can tell you the development of blooms through tracking the chlorophyll A development in a lake. And there's also great consistency of measurement between agencies and countries, which we don't have necessarily through lab measurements of chlorophyll, for example. Obviously, there are some sort of negatives as well, and that's particularly cloud covers the big issue. For cyanobacteria, there's still work to be done on algorithm calibration and validation. And for the phycocyanin, because it requires sort of hyperspectral data, it's only available for the larger lakes only. But there are some real synergies between citizen science and satellite data, and there's also potential for the two to be cross-validate each other as well. And so we're looking at those options as well. So I just want to do the final bit of the talk on drivers of blooms. I will need to be very quick, so I might have to skip some slides. This first graph just shows you the sort of abundance of cyanobacteria in lakes of different alkalinity type. And you can see that low alkalinity lakes don't generally have high levels of cyanobacteria, but they do occasionally. And as I said, you can get sometimes these shoreline accumulations of high risk events. But the bloom risk is really much greater in higher alkalinity lakes. Need to click on there. Something's gone wrong. I just need to. Right. Okay. Now, we often, there's a lot of discussion in the literature on whether phosphorus or nitrogen management is required. Well, some of the data from this European analysis highlights that actually management should still largely focus on phosphorus, because it's only really in lakes with excess phosphorus that you get these really intense blooms that exceed WHO guidelines. You do obviously in other lakes you do get high levels, but not so frequently. So there is some discussion about nitrogen limitation favouring cyanobacteria. But our analysis seems to show it's not nitrogen limitation as such, it's an excess of phosphorus that's really important to maintain these high abundances. Oh no. Sorry about this. I think my PowerPoint is crashing.
It's like it's loading, but just taking its time.
Okay. It's crashed, I think. So I'm going to just quickly start PowerPoint. Sorry about this.
All right, it happens.
Oh, can't see it there. Where's it gone? Maybe this one. Something's going wrong.
Laurence, you might have to stop sharing and then reshare for it to show up as an option.
Okay. Is it? I can see it now. Are you getting it?
We're still seeing your holds.
Can you see it now?
No.
Okay, I'll stop sharing and right, I think that stopped sharing.
Yeah.
Let's see if that works. How about that?
It's loading. Yeah, you're back at the start of that.
Right. Let me. Oh, that's lucky. Just that.
Well done.
Have we lost Laurence?
I think we might have.
I might have lost Laurence, so give him a couple seconds.
Oh, yeah. We lost.
Can you hear me?
Yes, we lost you momentarily, but we have you now. Great.
Can you still see my screen?
Yes, we're on the cyanobacteria versus TP.
Okay, great. Sorry about all this. The general trend is increasing levels as low as 10 micrograms per litre of total phosphorus, but then sort of plateauing out at about 100 micrograms per litre. But you can see this huge scatter in this relationship, largely due to other limiting factors. And so we've taken a quantile response to looking at this because of these other limiting factors, and I'm going to go just quickly through this. And so you can use quantiles, put a number of quantiles through this sort of very large scatter of data to sort of understand the sort of probabilities of cyanobacteria abundance. So this is sort of the maximum capacity, and then you can see the median response. And you can actually think of those quantiles as different likelihoods of exceeding. You can put a level, a threshold in of abundance, and then you can look at what's the likelihood of exceeding that threshold using these quantiles and measure the phosphorus concentrations for those likelihoods of exceeding those thresholds. And so that's what we've done, and we came up with this sort of summary of that does actually show you that even at very low levels of phosphorus, there are some chances that you will have risk levels above the WHO low risk threshold, and that those risks increase quite rapidly between 20 and 50 micrograms per litre of phosphorus, and then they start to plateau out after that. And so clearly we need to reduce nutrients quite a lot, but we also need to understand variability in the response. And we've been particularly interested in looking at climate change variability using a number of data sets across Europe. So these are long-term time series, 26 lakes. And we've been looking at the response to climate variables as well. And this just shows you the response to rainfall in these 26 lakes. They're very, very varied. This is cyanobacteria abundance on the y-axis, and then the rainfall levels along the x-axis. And you can see the correlation coefficient between them. You can see it's very varied. But if you look at short residence time lakes, you see a much more consistent negative, significant negative relationship. And we can just plot those, and you see actually a fairly strong relationship for rainfall in these short residence time lakes. That actually is stronger than the nutrient effect in these lakes. If we look at temperature effect, we don't see much of a temperature effect on mean cyanobacteria levels in lakes, but you can see from this box plot that if we look at the lakes in relation to sort of average summer temperatures, the sort of upper quantiles, so these high levels of cyanobacteria abundance are really really start to increase dramatically when temperatures start to go over about 17 degrees C in our waters. And so, again, some sort of quantile analysis is maybe more informative to understand these really peak bloom events. We've also looked at the sort of effects of multiple stressors to temperature, climate, and nutrients at a European scale. There's a couple of great papers by Jessica Richardson, a PhD student, on this. And she's done a really nice approach looking at how these stressors interact differently in different lake types. So this table just shows you different lake types and then the effect size of phosphorus, temperature, retention time, and whether there's any interaction effect. And you can see different variables are important in different lake types. So temperature is particularly important in the deep stratified lakes, and retention time clearly more important in the shallow mixed lakes, like we see at Loch Leven. And we do see some lake types where we see significant interactive effects. And you can create these models just not for lake types, but for individual lakes. And we have enough data at Loch Leven to create an individual lake model of responses to multiple stressors. And you can see this is a heat map of the chlorophyll response in Loch Leven, with very high levels of chlorophyll at high nutrient levels and low growing season rainfall levels. And so Loch Leven at its current status, we can get a picture of, we can use these models to understand what sort of level of nutrient reductions required to meet good status. But we can also use it, look at future rainfall predictions and see how that's going to affect how much nutrient reductions we're going to need to reach the good status target. So in the future, we are going to likely to need greater nutrient reductions to mitigate against climate change. So just to summarize, I've highlighted that it would be really helpful to have a more integrated approach to understand the real health impacts of algal blooms. We're beginning to get that in the US, and I think in Scotland we're going that way, but I think it would be great to start really pushing for that across the UK. Citizen science and satellite data can really help as part of that, developing new data sets to reduce risks to the public and animals, but also to understand the risks a bit further as well. And hopefully I've shown that these larger data sets now we can use them to understand how we can mitigate aspects of climate change, what levels of nutrient loads are required to mitigate those changes. And sorry about all those issues that we had, but I just want to thank a few people there at AGC who've contributed to this talk and open the room for questions.
Laurence, thank you so much. I have received somebody, yeah, thank you. The time may elapse. If for any reason it does, because I scheduled the meeting for 1 hour, so that's my fault and my apologies. If for any reason Teams cuts us off, may I suggest that we just reconvene immediately, because I know that there'll be many questions for Laurence. So I'll pass over to one of the first ones we've received, but yes, if that happens, my apologies, and let's meet immediately to resume where we left off. So I think we have our first show of hands. I'm just going to do it in the order that they are in front of me, so apologies if it's not chronological. If I start with Ellis, if you'd like to unmute and ask Laurence your question, please.
Hi, yeah, I think a very interesting talk. I just wondered about how applicable this is for eukaryotic algal blooms. So you mentioned euglena, which can be toxic, particularly for fish, and in the Americas mostly, and Primnisium in the Norfolk Broads, which has had effects.
Yeah, so I mean, our app is really targeting cyanobacterial blooms, but obviously, clearly, we can see now we begin to get a feel for euglenoid blooms. I think it is quite difficult for us to distinguish those with our just a simple visual photo. So I think it does need chasing up. But then, if these are issues for fish kills and things, then maybe it doesn't matter so much. I think we do need a greater understanding of that. They're often widely ignored, and so hopefully it can give us a bit of a picture on the scale of these different forms of blooms.
Thank you.
Thanks, Ellis. Could I next go to Hilary, please?
Oh, yes, that was great. Thanks for the stuff about the Bloomin Algae app. And I was just wondering if there was any thought of putting in something like CCTV round about popular areas, e.g. like Windermere or Loch Lomond to look for algae blooms, so you can have a real-time monitoring. I'm just thinking, like up here in Aberdeen, if I want to go out, I'll look at the Snowgate CCTV monitors to see if the roads are closed or open. It could be a thought.
Actually, it would make real sense for sites like Loch Lomond, South Basin, Windermere, these high tourist spots. I think that would really make sense. So yeah, I can talk to. The only issues with CCTV, I guess, there are some GDPR issues. I think these days, but because we have thought about putting them at the harbour at Loch Leven, and just there were some issues. We just didn't know how we'd get around some complicated issues associated with that. So it put us off. But I'm sure it is possible for landowners to do it.
Great, thanks.
It's a good idea.
Thanks, Hilary. Yeah, it is a good idea, actually. Linda, you have a question for Laurence, I believe. If you'd like to unmute and ask.
Hi, Linda. I haven't seen you in a long while.
Can you unmute me from the other side?
We've got you. Yeah, I think it's just that slight delay, isn't it? But yeah, we've got you now.
Okay. My question is on your sample size that you take. I wondered how much water you took for each sample that you analyze, because we have a system where you can take up to 50 liters and filter that in about 30 minutes. And I wondered if a larger sample size would be of any interest to you and your apps.
So the app doesn't, obviously there's no water sampling involved with the app at all. That's literally just done through visual analysis by an expert of the photo, just giving a expert judgment of whether they think it's a bloom or not. It's very rarely do we get samples analysed of those. The few sites that we do are the ones that SEPA and CH analyse, so we have background data on those, and occasionally the environment agencies will check a sample out. So we don't have that. So for our normal routine monitoring, though, we're taking half a litre or so of water, and you know, really, well, actually, we're only analysing usually, 10 mils of water or something like that in our inverted chamber for cyanobacterial counts. So I don't know how you would necessarily think about using larger volumes, really, or the need for that.
Or well, if you're doing it year round, so at times when the algae are not really blooming, but they're present in the water column. So our system that we have takes a 50-liter sample and concentrates all the organisms down to a liter, and then you could take your aliquots out of that liter and scan for your cells. But the question is whether it's of any use to you to have a 50-liter sample that's been concentrated.
From a cyanobacteria concentrations, for our sort of microscope counts, I don't think we need that much, because even in winter when we've got very low levels, usually a litre or so is enough, and we sediment down from a litre. But I don't know whether from a toxin perspective or whatever that would be useful. But certainly from a cell count, I think we usually can deal with a litre or so is sufficient.
Okay.
I don't know if Dave's got his hand up for a question as well, but I don't know.
Chris. Yeah, Chris, would you like to ask Laurence?
Thanks very much indeed. You talked about the role of phosphorus and so on in cell counts. What about triggers for toxin production? Because of course it's not just having the cells there; it's producing the toxins.
Yeah. So our lab, we don't generally do toxin analysis in our lab. We leave that to some other experts up in Aberdeen, Robert Gordon University, so we, from the data I've seen, I think between sort of 50% to 90% of blooms are toxin-producing blooms, and so from a risk management perspective, I think it's a reasonable assumption to assume that toxins are maybe being produced by that bloom. And because it's quite dynamic, both the bloom and the toxin production is quite dynamic. I think from a risk management perspective, you have to take that point of view. But from understanding toxin production, I think it would be great if there was more cyanotoxin analysis done routinely, so that we could actually start unpicking both the environmental controls of cyanotoxin production. I think would be great. And I know the groups like Linda Lawton's group, Christine up at Robert Gordon, who are doing a lot of work on that, and groups in the Netherlands as well. There's some, Mikhail Lurling's group and people.
Thanks very much.
I don't believe we have any further questions. Linda, your hand is still up. Is that just left over from before? You don't have an additional follow-up question for Laurence, do you?
I don't. I think it was just left over.
Yeah, okay. David, do you have a question for Laurence? Or you're currently... Yes, there we go.
Yeah, okay. Hi, Laurence.
Hi, David.
I was very interested in your app again. I must admit, I haven't used your Bloomin app myself. But, I mean, obviously it's telling you there's an algal bloom and you're trying to interpret what the algae are. Have you got any plans in the future for people to actually do some proper ground truthing of these blooms? Because I certainly been collecting blooms in the London area, and I usually get microcystis, but then suddenly I get what used to be called anabina, actually, blooms, and they look very similar, but when you actually get close up, there are subtle differences, but they probably wouldn't be shown in the photographs you were sent. But I think increasingly people are using microscopes, and I think you might find people would like to send you photomicrographs, which you will be able to then sort of identify those blooms down to at least the genus, if not down to the species.
Yeah, we do have one or two users that send us photomicrographs, actually. So they send us a lake shot and a microscope image, which is great, because then immediately you can be sort of reassured about the algae involved. So we would really encourage it, and certainly I'd be very welcome if many BPS members who have access to microscopes use the app to broaden the spatial coverage. But if they have access to microscopes and taking images, even better that they can submit a microscope image as well along with the sample.
Well, it's getting so much easier. I mean, as you know, you can just take the image with your smartphone and down if you've got the microscope, of course. But microscopes are getting so much cheaper, and I think it is something to encourage people to increasingly do, actually.
Yeah. Well, maybe with these old scopes we can encourage it. I don't know. I have a USB microscope as well that is fantastic, actually. You can get clearly see microcystis and Dolichospermum filaments very easily.
Oh, no, I thought so. Oh, a very interesting talk, Laurence, actually. Thanks.
Well, nice to see you, David. Hope Bromley's
okay.
I think it sounds like a great challenge for us with students as well. You know, if we do know that we've got sites near us where unfortunately we're having these blooms, you know, what a great way to get them out ground truthing as well. So yeah, good thought there. I've used it myself. Yeah.
And certainly the training, we decided we will want to focus the app. So it's for the general public to use, but we'd be very happy to focus training in the future where certain experts could get more out of it themselves. Yeah. You know, and so, as you say, submit close-up photos. We'd be very open to more verifiers as well, people who want to have a lake or a region that says, okay, if any records come in from this lake, I will go and check it as well. So that's fantastic.
And also, if these records can be entered up in the BPS algal recording website as well with photographs.
Yeah, well, we'd be happy to talk about automating that if needed from our, because we can send the data to other
that would
websites.
be good, actually. Yeah.
Fantastic. There's been a few kind of comments in the main kind of channel that we have. I haven't seen any particular questions there for me to pose to you, Laurence, on their behalf, but definitely some good ideas about you know, perhaps collaborations and the like. So do feel free to have a look at that, as well as other types of microscopical societies that are interested in the app as well. So before we kind of round up, does anyone else have any final questions for Laurence? That was just another fantastic thumbs up there from Hannah. Laurence, well, please go ahead.
Want
Sorry.
to email me, because I see some people have said get in touch. Well, if they want to email me, I've given them my email, and we can follow up after the talk.
That's fantastic. I'll just check if anyone else has said anything. No. Okay. Oh, can I send records from estuaries? Was one final question there, or is it simply freshwater?
They can be. We are focusing on cyanobacterial blooms as the thing that we can identify, but you know, it's worth having records. As I said, even the rejected filamentous algae records are now being used by Hannah in her PhD at Nottingham. So data that we thought wasn't really relevant is now actually of use for sort of thinking about managing filamentous problems, nuisance algae. So yeah, I don't see why not.
Okay, that's good to know. Fantastic. Well, Laurence, thanks again. Really, really interesting and great to see the application of it, you know, in practice. So yeah, I look forward to seeing the updated app, and I'm sure there'll be a lot of us popping up on there, bombarding you with photos.
Right.
Okay, thanks everybody. We end there, and we will be meeting again at 12:30, so quite soon if you want to have some lunch. But Ellis and Eleanor will be chairing that session on applied phycology. So yes, without